This is BIOS Daily News. Hunter Manning here today, reporting live on top news stories in the scientific world. Today we focus on a fascinating new discovery in the field of epigenetics, dealing with the microscopic aquatic rotifer. I'm going to turn it over to Kathy for a little background on this science.
Thank you, Hunter. I'm Kathy Quillian, and today we'll be focusing on the growing research of epigenetics. Epigenetics is the study of how behavior and environment influence gene expression. These changes in gene expression are extremely important because they are capable of triggering phenotypic changes in an organism without alterations to the DNA sequence. The Rotifer research we report on today focuses on epigenetic changes involving DNA methylation. This works by adding a chemical group to DNA, a methyl group, which blocks proteins that attach to DNA to read it and produce proteins encoded by the gene. Thus, the added methyl turns off the gene. Gene regulation is important when it comes to transposons. Transposons are repetitive elements of DNA that are able to move to various locations within the genome. In some circumstances, transposons have the ability of inserting foreign DNA into an organism's genome, as shown in this graphic. The scientific research we debriefed today recounts epigenetic marks discovered in rotifers, which enable them to suppress these potentially harmful transposons.
Thank you, Kathy. As we can see here, rotifers are small invertebrates often found in environments that are subject to extreme drying. Rotifers are known for their ability to incorporate foreign genes into genomic DNA. This is potentially due to their desiccation phases, in which their DNA breaks up into pieces and potentially renders it accessible to foreign DNA fragments during rehydration. Fernando Rodriguez and Irina A. Yushinova are the main contributors to the scientific literature discussed today. They are both research scientists at the Josephine Bay Paul Center for Comparative Molecular Biology and Evolution. Their collective works delve into deltoid rotifers and our transposable DNA as a whole.
DNA modifications used to regulate gene expressions and protect against transposons are typically involving C5-methylcysteine in eukaryotes. Belloid rotifers, however, encode an aminomethyltransferase, N4-CMT, captured from bacteria. This non-native DNA methyl group was horizontally transferred to belloid rotifers some 60 million years ago and has reshaped their epigenetic system, enabling them to silence transposons. The researchers in this article demonstrated that bacterial IMTACE established 4MC marks do in fact act as an epigenetic mark in belloid rotifer genomic DNA, gDNA, rather than the typical eukaryotic 5MC marks from C5 IMTACE. Here we can see phyletic distribution patterns of methyltransferases implicated in 4MC and 5MC genetic marks from the primary research literature. As shown, belloid rotifers and bacteria share the N4-CMT gene responsible for 4MC marks, while the 5MC mark appears to be absent in the rotifers. The researchers found that Adinetta vega belloid rotifers have one gene of bacterial origin, which was closely related to amino-IMTACEs of bacterial restriction modification systems, which provide bacteria defense against foreign DNA. The researchers also found that the rotifers lacked 5MC deamination signatures in genomic DNA that would typically be found in regulatory gene modifications in eukaryotes. They extracted genomic DNA from A. vega and introduced anti-4MC antibodies, which resulted in a positive reaction, thus suggesting the presence of 4MC marks. To examine the genome-wide distribution of modified bases, they used single-molecule real-time sequencing, which provides single nucleotide resolution, which enabled them to detect transposable element copy integrity at different DNA methylation densities. As shown in the figure, as the length of the TEs increased, so did the amount of 4MC DNA modifications, thus indicating that 4MC targets transposable elements for modification. In the graph shown, bar height indicates the average number of 4MC or 6MA SMRT counts, and error bars represent standard deviation for full, medium, and short transposable element copies. The absence of C5 IMTACEs, similarity of N4-CMT to bacterial N4-CMT tases, and the lack of 5MC marks support the researchers' hypothesis that cystacenes and belloid rotifers are modified at the N4 rather than C5 positions. Thus, it is N4-CMT horizontally transferred from bacteria that is responsible for the addition of 4MC epigenetic marks to DNA, not the C5 IMTACE typical of eukaryotes.
As supported by the research article, this piece of bacterial DNA joined with the rotifer's genome and formed a functional enzyme, creating a regulatory system that enabled rotifers to regulate genes and control jumping transposons. This extra layer of defense protects them against harmful mutations caused by transposons and thus increases their survival and ability to reproduce. The implications of this peculiar horizontal eukaryotic gene transfer may be future technology harnessing similar gene transfers in other eukaryotes, enabling epigenetics that could suppress transposons. One current related issue in the human body is the line-1 transposon, which has been linked to different kinds of cancer due to its potential to land on APC, a gene essential for making tumor suppressor proteins. If we can apply knowledge of the horizontally transferred gene that enables the silencing of transposons, we may be able to develop technology to silence harmful transposons in people.
This rotifer discovery illustrates the principle of evolution by natural selection relating to Module 1 of this course. Rotifers captured this bacterial gene about 60 million years ago, and because of the reproductive and survival advantage it offered them, rotifers with this gene were able to persist and pass it on to offspring. Thus, this horizontal gene transfer from bacteria, which induced a new epigenetic mark, enabled them to regulate potentially harmful transposons and was preserved and amplified over generations.
That is the BIOS Daily News for this Monday. Thank you for joining us today to learn about this interesting rotifer discovery. Don't forget to tune in next time. Have a great day.